Welcome. Welcome to the Science and Tech Podcast.
Yeah, we're so glad you're joining us today.
Absolutely. And speaking of fascinating topics, Alex, do we have a big one for our listeners today or what?
Oh, it's huge, Elizabeth. We are diving deep into something truly groundbreaking from DeepMind.
That's right. We're talking about AlphaGenome, an AI that, well, let's just say it's poised to completely revolutionize our understanding of genetics.
Yeah, it's pretty mind-blowing stuff, honestly. It's like cracking the dark DNA code.
Exactly. And we'll get into what that even means in just a bit. So buckle up, because this is a deep dive into the future of genomic science.
So, for decades, Elizabeth, like for 20 years, scientists have been grappling with this huge puzzle in our DNA. They called it junk DNA or dark matter.
Oh, yeah, I remember hearing about that. It's like, what's all this extra stuff doing?
Exactly. And the thing is, it's not a small part. We're talking 98% of our human DNA doesn't actually make proteins. So for a long time, it was just like, what is this doing here? But here's the kicker. Even though it doesn't code for proteins, mutations in this regulatory wilderness, this dark matter, are actually linked to some really serious stuff.
Like what, specifically?
Well, they've found connections to things like autism, cancer, and heart disease. It's not just inert, It's this vast non-coding region that, despite its name, plays a really crucial role. And for so long, we just couldn't quite figure it out.
And that's where things really took a turn, because get this, Alexander, everything changed just recently. On June 25th, 2025, that's when Google DeepMind, the AI powerhouse, they officially unveiled AlphaGenome.
Oh, wow. So this is like very new, bleeding edge stuff then.
Absolutely super fresh. And what they claimed in their blog post, it was just mind-blowing. They said this new transformer AI could, get ready for this, it could read 1 million DNA letters at once. And not just that, but it could predict over 32,000 molecular features across all major cell types.
32,000. That's an incredible amount of data to process simultaneously.
I know, right? And it could also instantly score how every single possible nucleotide change would ripple through that entire genomic landscape. I mean, within hours of them announcing this, Nature, Science, Stat News, they were all over it, headlining it as a, quote, potential Rosetta Stone for genomic medicine. It was just huge.
Wow, a Rosetta Stone. That's a pretty bold claim, but it sounds like it might just live up to it. And, speaking of how it works, there's a really interesting part about why this context window is such a big deal.
Ooh, tell me more. What's the secret sauce there?
Well, it's kind of like long-distance romance, right? Gene regulation often involves an enhancer, which is a DNA segment, sitting like 100,000 bases away from the promoter it activates. Earlier models, they couldn't see that far. They'd hit a wall. But AlphaGenome, it totally solves that memory crunch. It uses something called sparse axial self-attention.
Sparse axial self-attention. That sounds very sci-fi.
It does, right? But what it means is it lets each token, each piece of information, attend to really distant sites without, blowing up GPU costs. So suddenly, it has this massive 1 megabase, that's 1 million base context window. And that captures most of the enhancer promoter, insulator, and silencer loops that biologists have documented. It's essentially giving the network the real spatial cues that biology actually uses. It's huge for understanding the full picture.
Wow. So it's like, it's not just seeing the whole conversation, but it's understanding the nuances across that entire massive distance. But what's incredible, and what's even crazier, I think, is that it's not just good at one thing. You know how normally you'd train like a separate AI for every single experimental assay?
Yeah, totally. It's usually a bespoke model for each task, right?
Exactly. But AlphaGenome, it has this thing called a multitask head. So instead of like 100 different networks, it's one model that can predict a whole bunch of different biological features. It's really versatile.
Oh, so what kind of stuff can it predict then from this single model?
Well, it's pretty extensive. It can predict RNA output for over 1,000 different GTEx tissues, which is huge. It also predicts like 20 different chromosome marks. Things like H3K27AC, H3K4ME3, all those fun ones. Plus, it covers DNase and ATAC accessibility, which are key for understanding open chromatin.
Right, so it's not just the DNA sequence, but how it's actually structured and accessed.
Precisely. And it goes even further, predicting over 1,000 transcription factor binding tracks and even DNA methylation patterns. So yeah, it's pretty much a one-stop shop for a lot of these crucial genomic insights, all from one unified model. It's pretty mind-blowing when you think about it.
Yeah, it really is. And to achieve all that, Elizabeth, you can imagine the sheer scale of the data and compute power behind it. I mean, we're talking about training AlphaGenome on, get this, over 40 terabytes of mixed genomic data.
40 terabytes? That's an insane amount of data. What kind of data are we talking about?
Oh, it's a mix from all the big ones. ENCODE, roadmap epigenomics, GTEx, and even a whole trove of single nucleotide CRISPR screens. So they basically just fed it everything. And the compute budget was comparable to AlphaFold, which gives you an idea of the resources Google DeepMind poured into this. We're talking something like 25,000 TPU V5e core hours.
Wow, that's a serious investment. So how did they, teach it to understand all this? Was it just throwing data at it?
Well, they used a really clever approach called pre-training and fine-tuning. So initially, they pre-trained it as a masked language model, which is like teaching it the basic syntax of the genome, how the letters fit together. Then they fine-tuned it on those functional tracks you mentioned, the RNA output, chromatin marks, and so on, to give it the actual biological semantics, like what those sequences actually mean and do. It's a really smart way to build a robust model.
That is really smart, actually. It's like teaching it to read before teaching it to understand poetry, right? And, with all that sophisticated training and massive data, it has to perform well. What were the actual benchmarks like? Did it really live up to the hype?
Oh, it absolutely did.
Okay, spill, because how much better are we talking?
So when it came to gene expression variants, AlphaGenome explained 66% of it, which is like double Informer's 33%. That's a huge leap. And for splice site effect prediction, its AU-ROC was 0.93 compared to Splice AI's 0.82. Those are some serious improvements.
Wow, doubling the explained variance is mind-blowing, especially for something as complex as gene expression.
Exactly. And then for ClinVar pathogenic variant recall, which is, super crucial for identifying disease-causing mutations, AlphaGenome hit an incredible 92% in the top 1% range. The previous best was only in the low 70s. That's a massive jump in accuracy where it really counts.
92% for pathogenic variant recall? That's just incredible. It really drives home the potential clinical impact of this.
Right? And here's another thing that really impressed me. Cross-species generalization. They found that for primate genomes, AlphaGenome retained over 80% of its accuracy. That's like a first for such a large context window, too. It shows it's not just memorizing human data, but actually learning transferable biological principles. That's pretty groundbreaking.
So after hearing all those impressive benchmarks, I'm thinking, okay, that's great on paper, but what about the actual, real-world impact? How is this actually being used?
That's the million-dollar question, isn't it? And honestly, the case studies are just as compelling, if not more so.
Lay it on me.
Okay, so first up, rare disease triage. Genomics England, they actually fed AlphaGenome scores into their massive 100K genomes pipeline. And get this, pilot runs flagged new intronic variants behind congenital heart disease that previous methods like SNF and CAD had totally missed.
Whoa. So it's like catching things that have been, hiding in plain sight? That's huge for rare diseases where a diagnosis can take years.
Exactly. Think about the time and suffering that could save. And it's not just about finding what's hidden, it's also making things more efficient. For cancer driver hunting, Memorial Sloan Kettering, they used AlphaGenome scores, the A scores, to pinpoint a KRAS enhancer 40 kilobases downstream in pancreatic tumors. And when they knocked that out using CRISPR-I, it cut oncogene expression by 60%.
60%? That's incredible. So basically, it's helping them identify potential therapeutic targets with unprecedented precision.
Absolutely. It's almost like having a spotlight for the critical genetic levers. And speaking of CRISPR, it's also being used for safety. A startup called Crux Bioengineering, which designs base edit repairs, uses AlphaGenome to predict off-target regulatory hits in silico.
Oh, so before they even go into the lab.
Precisely. It basically trims their wet lab screening costs by half because they can filter out the problematic designs virtually.
That's really smart. That's a massive cost saver, and it makes the whole CRISPR design process so much more efficient and, safer. Are there any other unexpected applications?
Yeah, there's a cool one in synthetic biology. The iGEM teams, the student synthetic biology competition, they're iterating promoter libraries computationally using AlphaGenome. This means they only need to order the top candidates for lab validation, which again saves so much time and resources.
Wow. So it's not just about understanding existing biology, but actually designing new biological components more effectively. That's truly expansive in its application, isn't it? From diagnostics to drug discovery to building the future of biotech. What's really striking about all of this, beyond just the incredible power, is the approach DeepMind is taking with Access.
Oh, you mean the API?
Exactly. They've actually mirrored AlphaFold's playbook, which I think is just brilliant. DeepMind launched a free, non-commercial API for AlphaGenome on GitHub. So, users can submit up to 5,000 variants per job for free. If you need larger batches, they run them at cost.
That's huge for the wider scientific community, right? I mean, not everyone has access to that kind of compute power.
It truly is. And what's really, really crucial, I think, and something that speaks to the transparency aspect, is that the API returns what they call attention maps.
Oh, I remember reading about those. What exactly do they do?
So these attention maps, they essentially highlight which specific DNA segments actually drove the prediction. It's not just a black box spitting out a result. You can see why AlphaGenome made that prediction. And that, Alexander, is gold for hypothesis generation, for, deeper research, and especially for regulatory scrutiny in future diagnostics. It builds trust.
Yeah. And I think that transparency, that ability to see why it's making a prediction, it's so important because, predicting disease from raw genome files, that definitely raises some alarms for people. There are significant ethical frontiers here we need to consider.
Absolutely. Privacy is a huge one.
Right. And potential misuse, too. I mean, we're talking about incredibly powerful technology here. But DeepMind, to their credit, they've really thought about this. They have a policy that bans gain-of-function pathogen work, for example, and they require encryption for human data.
That's good to hear.
And they've even seated an independent AI genomics ethics board, which is chaired by Nita Farhani. Plus, regulators at places like NIH and EMA, they're already drafting rules to ensure that wet lab validation actually precedes any clinical decision based on AlphaGenome scores. So it's not just a free-for-all.
It feels like they're trying to get ahead of it, which is crucial for something this transformative.
Exactly. And looking ahead, their future roadmap is just as ambitious. Actually, they're planning a single-cell mode for even finer precision, cross-species transfer for preclinical work, and even exploring neuromorphic hardware. And critically, they've got clinical pilots coming, like Oxford testing AlphaGenome-guided variant triage in newborn intensive care units. Yeah, it's really pushing us to debate genomic privacy before this tech becomes completely mainstream. But ultimately, it could really illuminate that silent 98% of DNA we've been calling junk, accelerate rare disease diagnoses where every day literally counts, and slash drug discovery costs. It's a whole new era, honestly.
Yeah, it really sounds like a new era, doesn't it? I mean, to think of all that dark DNA finally being illuminated, accelerating diagnoses, cutting drug costs, it's just mind-blowing.
Absolutely, Elizabeth. And, the fact that it's accessible through a free API, really democratizing research like never before, it's a game changer on so many levels.
It truly is. Well, that brings us to the end of another fascinating discussion here on the Science and Tech Podcast.
Indeed. Thank you all so much for joining us for this deep dive into DeepMind's AlphaGenome. It's been an incredible topic.
Yes, thanks for tuning in. We'll be back next time with more exciting breakthroughs from the world of science.
Until then, take care, everyone.
Bye for now.